Hello, everyone. I hope I'm back on. Is everybody there? Is anyone there?
Yeah.
Okay.
Yep.
Yes.
Okay. I've never had that happen before. Lost the internet right before the meeting was supposed to start. We're not going to spend any time on the rest of the material left over from the last PDF that we covered. So we're not going to talk about calcitonin or any of those kind of things. Can you see the entire slide?
Yes.
Okay. What we're going to talk about today, well, first of all, an update on the exams. I haven't even gotten through halfway through the exams. Turns out grading them is a bit more time demanding than I anticipated. Plus I had some logistical problems initially. I'm adding more comments than I typically do on the exams, particularly the ones that are written in PowerPoint or sorry, in Word and saved in Word. I don't know exactly what I'm going to do with the PDFs because the other alternative, because I can't actually modify it. So we'll see. Thus far, the grades are lower than what they have been in the past, but that's not too surprising. I told you I was going to be more demanding this time. My favorite questions on that exam were 5 and 6. If you think about 5, what it is a positive feedback loop where presence of X stimulates or de-represses the X operon, resulting in transcription and expression of the X gene product, which in turn produces more X product, and also the equivalent of CAP, catabolite activating the lac operon to maximal expression, catabolite activator protein, CAP. So you've got both processes going full steam ahead for the production of X. But like most things, too much of a good thing is too much. So we need a mechanism to tamp it back down, to cut down or shut off the operon. and that's where the gene Z comes in. That when there is sufficient X present, it will get converted into Z, and then Z will interact with a dormant repressor to bind to the operator and shut the whole thing down. Now, it creates a bit of a problem in that it's conceivable that you would end up producing very little X before it gets converted into Z, and then shut the whole process down very early in the process. And I want you to think of ways, and we'll come back to this at the end of the presentation. I want you to think of ways that you could maximize expression of the X gene and minimize expression of Z, so that you would build up an excess of X before Z starts to kick in. And we'll talk about that at the end of the class. Later on today, I will be sending you the key that I'm using in grading the exam. The answers on the key are what I think are the best answers, the most straightforward, the easiest answers, but they are not the only answers. There are other answers that I found acceptable. So don't panic if you don't have exactly what is on the answer sheet, but I'll be sending that to you later on this evening. Okay. Now, the end of the last presentation was to go over different ways that genes can be regulated. And if you think about any mechanism to regulate gene expression, Mother Nature is already doing it. But we're going to talk about one in particular today, and that's alternative RNA splicing and how that can be used to regulate phenotypic outcome. Okay, we're going to talk about sex determination in Drosophila. We already talked about how sex is determined by the ratio of X to autosomes. If the ratio is 1 to 1, that is 2 autosomes, 2 X's, the end result is a female phenotype. If the ratio is 2 autosomes to 1 X, that is a 1 to 0.5, then the outcome is a phenotypic male. And that implies in and of itself that there must be some kind of counting mechanism to determine the ratio of autosomes to X chromosomes. And what we're going to talk about is exactly those ratios. Now, I've updated the PowerPoint presentation from what I sent to you all. So some of the things have changed. There's some more additional information on it. I'm hoping that I am recording this, but since I got kicked out, I have no idea if I'm actually recording it or not. So if I am recording it, I'm going to try and figure out how to upload it to YouTube, in which case I'll send you all an email to let you know that it is present on YouTube and what to look for. Okay. How do I get rid of all the photographs? Part of my screen there, that's, no. Part of my screen is obscured by images, and you weren't supposed to actually be able to see me, but apparently I guess you can. Can you?
Yeah, we can.
Oh, shucks. Well, that wasn't supposed to happen. Oh, well, it is what it is. There are 3 main components in the initial determination of sex in Drosophila, 3 main genes. There is sisterless, which is found on the X chromosome. There is deadpan, which is found on the autosomes. And there is a gene called sex lethal. It's the competition between sisterless and deadpan that determines whether or not sex lethal gets transcribed. Sisterless actually inhibits the effects of deadpan. The function of deadpan is to inhibit transcription of sex lethal. So if there are more autosomes present than X chromosomes, deadpan, the inhibitor, is going to prevail, and it's going to prevent sex lethal from being expressed. If there is an equal ratio of X to autosome, Sisterless is going to interact with deadpan, inhibiting its effects and allowing for sex lethal to be transcribed. As I say, the autosomes, the inhibition factor to repress expression of sex lethal, is referred to as a gene called deadpan. Sisterless is found on the X chromosome. Its function is to inhibit the effects of deadpan. So if you have an even ratio of X autosomes, the inhibitor sisterless dominates and allows sex lethal to be transcribed. If the autosomes dominate, 1X to 2 autosomes, the repressor, deadpan, found on the autosomes, dominates, and it will inhibit the transcription of sex lethal. Expression of sex lethal results in a cascade of events that's going to result in a female phenotype. And this involves differential splicing. We're going to talk about several different instances where differential splicing plays a critical role in this process. Sex lethal is a differential splicing factor for the next gene in the cascade, referred to as transformer. So sex lethal is going to allow for the presence of the protein, sex lethal, is going to allow for an alternative splicing event of the transformer gene. The transformer nuclear RNA, nRNA, the unprocessed form, is actually expressed in both sexes. But it's how it is processed, how it's spliced, that determines whether or not the cascade is going to continue to be female or is going to become male. The alternative splice form, involving sex lethal, results in a female-specific transformer. Sex lethal binds to the TRA, nuclear RNA, preventing spliceosome formation on 3' general splice site. A second, less efficient splice form is formed, utilized. The female TRA messenger RNA lacks an exon that contains a stop codon. The male is the default state, where the exon remains in, and so you have premature termination of the transformer protein, resulting in a non-functional transformer protein. Only female-specific TRA messenger RNA makes a functional transformer protein. The transformer protein itself is a splicing factor, alternative splicing factor. The next gene in the cascade is referred to as double sex. The TRA protein, if a functional one is present, results in a splicing of the double sex gene, which will permit female development, female morphology, female development. If you have a non-functional TRA protein, you don't get the alternative splicing occurring, the result is a male phenotype, resulting in the male-specific double-sex protein. So a functional TRA transformer modifies the double-sex mRNA in such a way that it produces a female-specific double-sex protein. Absence of a functional TRA transformer protein, the default state is to produce a double-sex messenger RNA, which is male-specific. In each case, double sex is a transcription factor. It's going to turn on some genes, turn off other genes. The female-specific double sex activates female-specific genes. For example, yolk proteins. And it's also, equally important, going to inhibit male-specific genes. So you're going to turn on female-specific and turn off male-specific genes based upon the double-sex protein. Male-specific double sex inhibits the same female genes that the female-specific one was turning on. It's going to inhibit those and instead is going to turn on the male-specific genes. So in this way, alternative splicing, first you have a counting mechanism to determine the ratio between X chromosomes and autosomes. That determines whether or not sex-lethal protein is transcribed. If it is transcribed, you have a 1-to-1 ratio of autosomes to X chromosomes, which is going to result ultimately in the female phenotype. If autosomes dominate, you're going to inhibit the transcription of sex-lethal. So you're not going to produce a functional transformer protein. The default state is to become male double sex. Functional transformer, because of the presence of double sex, results in a presence of sex-lethal. Transformer is modified by the sex-lethal, different alternative splicing, resulting in a transformer that is female-specific, which will cause double sex specific protein. This is what you need to learn. Basically, this is a summary of the whole cascade. Can you see the whole thing?
Yeah.
Okay. I can't tell what you all are seeing. So, but you have a counting mechanism, X to autosomes. That determines whether or not sex-lethal is going to be transcribed, which is the pink on the left. If sex-lethal is transcribed, it produces a female-specific alternative splice factor. In the absence of transcription of sex-lethal, no sex-lethal is going to be produced. The next gene that gets turned on is transformer. It's expressed in both males and females. If you have sex-lethal present, the transformer messenger RNA is going to be processed in such a way as to produce a female-specific protein. In the absence of sex-lethal, you have the default state of transformer, which is a non-functional protein. Then you express double sex. It's expressed in both sexes. In the presence of a functional transformer, it will splice double sex in such a way that it becomes a female-specific transcription factor. In the absence of a functional transformer, the default state is the male-specific double sex. And the female double sex is going to turn on female-specific genes, turn off male-specific genes, and the default double sex is going to turn off female-specific genes and turn on male-specific genes. Are there any questions about this material?
Is there a sex-lethal for males, or is it just the absence? Is it just the absence for all of them except for females?
Yeah, occasionally you get what we refer to as intersex. If you have some sex-lethal activity, a hypomorph, but if it's a complete null, the sex-lethal would result in the default state, which is males. So all the progeny would be male in that situation. Double sex is called double sex because depending upon where the mutations are located, it will either affect the female-specific protein or the male-specific protein. So you can get either, again, depending upon where the mutation occurs. Other questions? Yeah, go ahead.
Okay. So in a normal version of this with no intersex or anything like that, the only time that you have something that's male specific is at the end between double sex and the phenotype?
Yes. It's only the double sex that produces a functional protein that is male specific.
Okay.
The others simply produce no functional protein. Sex-lethal doesn't get transcribed, so it doesn't get produced. Transformer is transcribed and translated, but in the absence of sex-lethal, the splicing pattern is such that it produces a non-functional transformer. Make sense?
Yes, thank you.
Any other questions? Because I can't really see your all's faces, I don't get very much feedback unless you all speak up. So questions and things like that are appreciated. Next, we're going to move on to what is arguably the most important event, development in the field of molecular biology / genetics, probably in the last decade. Oops. Now I've got to get out of this PowerPoint. Oh, no, I don't. Still part of this PowerPoint. Okay, I want to give a little pitch for investment in basic science research. A lot of politicians in particular think that investment in basic science research is largely a waste of money, and they're right, most of the time it really doesn't result in any kind of usable product, although it does typically generate information that's useful, for example, in treating cancers and various other diseases. We are heavily reliant right now upon scientists in particular, but also physicians, especially physician scientists, to try and get us out of this disaster pandemic that we are suffering under with the coronavirus. We're about a month, 2 months late in actually responding, but the scientific community, both in private industry as well as in universities, are going full out now to try to conquer this disease. But I would point to 2 instances where investment in basic science research has paid off huge. The first is trying to understand why bacteriophage were restricted into which bacterium they can infect. That led to our discovery of restriction enzymes. In the absence of restriction enzymes, it's highly unlikely we would have the biotechnology industry that we have today. It was the pillar, it was the foundation. It was what allowed the multi-trillion-dollar industry in biotechnology to evolve. And we're going to talk about the other one that I think a modest investment has resulted in huge profits, if you will. And that's the CRISPR-Cas9 system. Investigations into CRISPR-Cas9 were basic science research. There was no promise whatsoever of anything at the end of the rainbow. It was curiosity. We've talked about how bacteria are very economical. They do not tolerate a large amount of surplus DNA in their genome. They seek to get rid of it as quickly as possible. But in sequencing a lot of different bacterial genomes, they realized there was a peculiar section of the genome that had palindromic repeats interspersed throughout the genome. People didn't understand what they were doing there. Because they weren't immediately eliminated, they assumed it must have some function. And that ultimately is what led to our understanding of CRISPR-Cas9. So we're going to now start talking a little bit about CRISPR-Cas9. Come on. It's a selective RNA-guided genome editing tool. I'm sure you all have heard a great deal about it. CRISPR stands for clustered, regularly spaced, short palindromic repeats. You could see why we call it CRISPR. It's a prokaryotic immune system. If you'd ask anybody 15 years ago about a prokaryotic immune system, the best they could come up with was probably restriction enzymes. But now we know that there is another system which protects from foreign elements such as plasmids and bacteriophage or bacterial viruses. Not only that, it's an acquired autoimmunity. No one especially ever thought that there would be an acquired immunity in bacteria. It was a real shock, but it turns out to be an extremely valuable tool. It is somewhat analogous to eukaryotic RNA, and it is an RNA-based targeting system. It's found pretty prevalently in unicellular prokaryotic, if you will, organisms or organisms without a nucleus, 40% of sequenced bacterial genomes, as last I looked, contained CRISPR elements, while 90% of the archaea contained it. So it's a very ancient system. Contains short DNA repeats with short segments of spacer DNA. And the spacer DNA is derived from previous exposure to either bacteriophage or plasmid. So things that had infected the bacteria before, if they survive, they actually take parts of the genome of the bacteriophage or the plasmid and insert it into this system, where there is the unique sequences from the invading pathogen interspersed with these repetitive elements. Spacer, as well as the palindromic DNA, gets transcribed upon infection. There's a trans-acting RNA, tracer RNA. I don't expect, we're going to go through a little bit of the history and how it actually works in bacteria, but I don't necessarily expect you to remember the specifics of this. I'm more concerned with the modern utilization of it in investigative labs, gene editing, things like that. And we'll talk about that. Cas9 is CRISPR-associated proteins. The easiest, the simplest, there are multiple different examples of this, of the equivalent of different Cases. Cas9 turns out to be the simplest. It's only a single polypeptide. Many of them have multiple different polypeptides. The reason you always hear about CRISPR-Cas9 is because it is the simplest and the easiest to manipulate. In wild type, the Cas9 has a nuclease, which cuts the spacer and repeat RNA into what's referred to as cRNA. Tracer RNA base pairs with the cRNA. Complementary to cRNA, guide gets cleaved. This is a simplistic illustration. You have Cas9 genes that are independent of the CRISPR system. You have a leader sequence prior to the palindromic repeats. They're all identical. The palindromic repeats are all identical. The spacer's DNA, however, are unique, and they are each derived from different segments of the invading pathogen, be it a plasmid or a bacteriophage. Cas9 is a nuclease. As I said, it has two active DNA cutting domains, two active nucleases, one for each strand of the double helix. Either or both nuclease domains can be disabled, and Cas9 still finds target DNA. As we talked about, many of the transcription factors, various different proteins, have at least two different domains. One recognizes DNA, and the other is where the business happens. In this case, it's a nuclease. But you can disable both nucleases or either one, and Cas9 still has the ability to bind to the targeted DNA. In the lab, we have combined tracer RNA and spacer to generate a single guide RNA, sgRNA, or a guide RNA. The single guide RNA plus Cas9 finds and cuts the target DNA. The single guide RNA plus Cas9 allows for gene editing. It's been demonstrated to work in organisms from yeast to monkeys to humans. Initially, it was shown in non-viable human embryos, that is, human embryos that got destroyed. But as I'm sure you probably are aware, there have actually been some human babies that have been generated, particularly in China. The researcher who did that, however, is in jail now because they violated ethical standards. Cas9 protein, should say Cas9, not CRISPR, can be modified to replace the nucleases with virtually any other active domain that you want. It can be transcription factors, can be activating or silencing genes, that is methylases, acetylases, deacetylases, you can replace it with virtually any protein you can think of, including GFP. There are large libraries of single guide RNAs that have been generated, mostly by industry, but also by university labs. If you didn't know, there's a huge court case going on, a huge patent dispute. Two different, very large labs are competing for the patent rights of this technology. You can see why, because the commercial application of it could reap millions, multi-millions of dollars of revenue. Just to compare and contrast a little bit, CRISPR-mediated interference as opposed to eukaryotic RNAi interference. Foreign DNA, formation of the CRISPR complex. The nuclease comes along, cleaves, in the bacterial case, the invading DNA, chops it up, gets it degraded, and then the bacterium can survive. So it attacks DNA. That's the main difference between the two. In the case of RNAi, it is also RNA driven, but instead of attacking genomic DNA, it attacks messenger RNAs. RNAi acts by either, if it's complete complementarity, can result in degradation of the targeted messenger RNA. If it's incomplete complementarity, it will still bind to the target RNA and block translation. So what advantages or disadvantages could you think of in terms of CRISPR versus RNAi? Modifying the DNA as opposed to knocking down translation product of an RNA.
Modifying the DNA would probably be a little bit more permanent.
It is a more permanent effect, yes, particularly if you're talking about germline. Thus far, CRISPR-Cas9 has been most effective in treating blood-borne diseases such as sickle cell anemia, where it's relatively easy to take out the blood cells, make the modifications, and inject them back into the host. There have also, though, as I talked about, been instances where in vitro fertilization coupled with CRISPR treatment of cells can result in an individual born with the modifications throughout all of their cell types caused by CRISPR-Cas9. So CRISPR-Cas9 causes a permanent change to the DNA. How do we replace, say, sickle cell anemia? How do we replace the mutant sickle cell anemia with the wild type? We've talked about how to target it. We've talked about nuclease activity, cutting up the DNA, but then how do we repair it? Does anybody know?
Could you somehow add the wild type DNA to it?
Yes. What you're going to do is to add a wild type copy at the same time you're introducing CRISPR-Cas9. Because there's been damage, because you cut up the DNA, the host cell, the cell that's being treated, is going to try to repair the damage. One way of repairing it is through homologous recombination. And if you've provided a relatively short segment of DNA, which will rapidly diffuse, it is more likely to use the rapidly diffused complementary wild-type DNA in homologous recombination to repair the breaks in the DNA. And so that's how you can repair it. Now, the efficiency is pretty good, and it's getting better all the time, but you nevertheless need to screen to identify successful replacements before you start injecting them into individuals, which is a fairly simple thing to do. You know the sequence in the region. You just PCR amplify, sequence it, and determine which clones have successfully replaced the mutated copy, this mutant sickle cell anemia, with the wild type. And the same is true of all other genes. So it relies upon homologous recombination. Now, there's recently been some clever approaches involving use of RNA that has the correct sequences, as well as a reverse transcriptase as part of the Cas9 CRISPR complex. The complex delivers the RNA with a reverse transcriptase to the site of the gene that you're targeting. Reverse transcriptase then makes double-stranded DNA, which is right in the vicinity of where the damage has occurred. So you're not depending upon passive diffusion, but you're actually delivering the targeted, corrected DNA to the site. It has increased the efficiency and reduced off-target effects. What's the advantage of RNAi? What advantage could you conceive of in the case of RNAi? Silence.
It the fact that it is not permanent could also be an advantage.
How so? You're right, but how so?
So if you need to change something, but you only, I don't know of any specific examples, but if you need to change something like in a mother, I guess, but you don't want the child to be born with those permanent DNA changes, then the RNAi could potentially give you that not permanent change.
Yeah, the key is it's transitory. And so RNAi, let's say you want to look at the effects of an enzyme during metamorphosis in Drosophila, during the process of going from a larvae to a pupa to an adult fly. But you only want to look at it during that stage, not before, not after. So you can drive RNA expression using the appropriate Gal4 UAS systems so that RNAi is only transcribed during a short portion of development. So you will only be knocking down the target for a short period of time during development. Once that time has passed, once that Gal4 stops being transcribed, you stop transcribing the RNAi, everything should go back to normal. So you can have a pulsed knockdown of any particular protein you want to using RNAi. So each has its own advantages. RNAi, in many ways, is very advantageous in studying developmental effects because you can be very selective in what tissues or what stages you're knocking down a particular protein. CRISPR-Cas9, in terms of gene therapy, is by far the best way to go. But the changes are permanent. Once you have made those changes, they will be perpetuated forever. So you can't have a transient effects. So RNAi, from an experimental point of view, is by far the more valuable tool. What's the downside of both of them?
Could there be an issue of ethics?
Well, there certainly is an issue of ethics, yeah, especially with CRISPR-Cas9. And it's one that we're just beginning to try to deal with. It's not trivial. Literally, the era of developmental babies are upon us. And so there's an awful lot of ethical issues involved. And we can explore some of those a little bit. No, the big problem, when RNAi was first discovered, pharmaceutical companies went gaga. They thought, oh, here's our cure for HIV. Here's our cure for lots of different diseases, especially RNA viruses. Boy, could we use it now for the coronavirus. The problem is that RNAi acts by one of two mechanisms. If the complementarity is perfect, then it causes degradation of the messenger RNA. If there is sufficient complementarity for it to bind, but the complementarity is not perfect, Instead, what it does is to inhibit translation. Unfortunately, it only takes a fairly small sequence to form complementarity. And so they discovered that RNAi had an awful lot of off-target effects. It wouldn't just knock down the targeted gene, but other genes would also be knocked down. And it turned out to be too dangerous, too deleterious to try to use it as a therapeutic agent. CRISPR-Cas9 also has some off-target effects. It's not nearly as bad as RNAi. And the technique I talked about delivering the RNA to the site, apparently significantly reduces off-target effects. So CRISPR-Cas9 is getting much better as the technology evolves in minimizing off-target effects. But they both have those caveats that need to be kept into consideration. One of the things of using CRISPR-Cas9, we talked about you need to confirm successful transformants, but you also need to look at genes that have similar sequences to your guide RNA to see if the CRISPR-Cas9 by mistake made cuts in an off-target RNA. Because if so, you don't want to put it back into a human being. Yes, there's a lot of ethical questions. As I say, especially with CRISPR-Cas9, RNAi has proved to be too hazardous to use as a therapeutic agent, which is too bad. CRISPR-Cas9, however, does anybody remember the story about the Chinese scientist? Well, in a research lab in China, in collaboration with an American collaborator, they knocked out one of the receptor proteins that's required for HIV infection in embryos. The concern, apparently the father was HIV positive, the mother was not. The concern was that maybe the father's HIV would infect the developing embryo. So they knocked out one of the receptors that's required for HIV infection. They did so without any kind of ethical oversight for doing these kind of experiments. It's something of a prophylactic. So they modified this infant's receptors, which made them more susceptible to other diseases, as not in a curative way, but in anticipation of potential problems. And so it raised a lot of concerns. There was never any ethical oversight. The Chinese government eventually arrested him, and I think he's serving several years in jail in China because of the lack of ethical concerns or behavior on the part of the researcher. But we literally are at the point where we could probably, if you wanted to have all your children be redheaded, you could do that. If you wanted them to have blonde eyes, you could do that. If you wanted them to have an extra round of cell divisions, in the case of muscle fibers, so you double the size of the muscles, you could do that. There are breeds of cattle now that are mutant that result in an extra round of cell replication in the muscles. So they end up being much bigger beast, which if you're selling beef is profitable. Same has been true of salmon and some others. And we know what those genes are and we know how to modify them. So there's a tremendous amount of ethical questions that haven't been resolved. And why is CRISPR better than RNAi? Well, it depends on what you're trying to do with it. So say, CRISPR targets DNA, RNAi targets the RNA. CRISPR makes a permanent change to the DNA. RNAi results in a knockdown of gene product. There are other approaches to genome editing that have been around for a while. Zinc finger nucleases. Zinc finger is one of the motifs that is involved in binding to DNA. So you take the DNA binding domain and you replace the activation domain with a nuclease. The zinc finger is specific for specific sequences. This has been used for a while. However, it's a game of trial and error, modifying the zinc finger so that it recognizes the sequences you want it to recognize and not other sequences. So it's a very timely, takes a lot of time and a lot of expense to try and develop zinc fingers. But once you have them, they are very specific. There's no off-target effects. TALENs, transcription activator-like effector nucleases, are also incredibly specific. It's a protein that recognizes specific base pairs, sequence of base pairs. The problem is that to recognize a single base, it takes 33 to 35 amino acids. So you end up with this huge, relatively speaking, a huge protein to recognize a relatively short sequence of DNA. But TALENs have been used. We have a researcher in our department, Sadie, who has used TALENs to modify genes in zebrafish. She did this before CRISPR-Cas9 became available. They're very target specific, tend to be, but again, both of them are time consuming and difficult to work with. CRISPR-Cas9 is simple. If I were still doing Drosophila DNA work, I would probably be using CRISPR-Cas9. Almost any lab can use this, as long as you can inject the CRISPR-Cas9 constructs into the organism that you're interested in modifying. As I said, the nuclease portion of Cas9 can be modified. You can replace the nuclease with virtually any other motif that you want, be it a transcription factor, be it something that's going to model chromatin, or be it something like GFP. Any questions?
Could you use CRISPR on like the BRCA gene or something like that?
Yes. Yes, you could. There's, as far as I know, there are no restrictions on genes that could be modified. Part of the problem with a lot of these is that in adults, you have to have a mechanism for getting rid of the existing mutated cells. The BRCA, you could generate cells that would be highly unlikely to develop into cancer, but you'd still have a reservoir of other cells present that would have the mutation. And so the question is, how do you get rid of that reservoir of cells? Part of the reason that blood is the easiest to work with is that we have been doing bone marrow transplants for a long time. We know how to destroy the bone marrow and then be able to replace it with the modified cells, so that you have stem cells that then have the modifications and will repopulate the bone marrow. You would have to segregate. Bone marrow is not simply one single blood cell type, but you'd have to be selective in what bone marrow you would use to replace. But you could replace the defective cells with the modified and corrected cells. But yes, you could use it on BRCA. And you could use it to have offspring that were free of the mutation. Because in the case of offspring, you're going to be starting with the single nucleotide, the pronucleus. So that would be fairly easy to ensure that the entire organism was free of those mutations. But in an adult, you've got the reservoir of cells that also have those mutations. And in the absence of coming up with a mechanism to destroy those cells, it's not going to be a terribly effective treatment. Does that make sense?
Yes, thank you.
Any other questions? It still really is a game changer, not just in humans, but in an awful lot of organisms, where we know modifications we can make to particular crops, for example. There's a gene that's involved in causing tomatoes to decay. They've already modified them in such a way that they've turned off those genes, but there may be similar genes that may be involved in something besides shelf life of tomatoes. You may get larger tomatoes, or you may get sweeter tomatoes, and various things like that. All those kind of modifications are going to be relatively easy to do once you've identified the genes to be targeted. So CRISPR-Cas9 is not just for humans, it's for all kinds of organisms. GMOs are a big issue in Europe, genetically modified organisms, which is silly. We've been modifying organisms since we first started agriculture through selective breeding and things like that. But now we have the tools to go in and selectively modify and not be dependent upon hybrids between different species in the case of plants and things like that. Ultimately, I think Europe's going to have to give up their border wall, if you will, against GMOs, because GMOs are going to be so much more successful than our existing strains. We need to maintain existing strains because we don't want to become a monoculture. A big part of the Irish famine was the result of the potatoes all being of the same strain. Once a fungus got in that could infect, it infected all of the potatoes. None of the potatoes, because they were all the same strain, they were a monoculture, had any resistance to the pathogen. And so we need to maintain at least some stock of wild type plants that could potentially have resistance to various different pathogens, both those that we are aware of and those that we are not aware of. You may or may not know, if you like wine, all vineyards, or virtually all vineyards across the world, the root is actually from American grapes.